I guess the psychology of drought stress seemed to attract some attention. Apparently some people, I won't name any names, thought maybe it should have been physiology, and indeed it probably should be. It's more about, do plants believe in fables, I guess, and that's where I wanted to start. So I did want to start with this. Did you know there's a video you can watch online? I haven't watched it, but it's called Plants Behaving Badly. Apparently it's about orchids and carnivorous plants and things like that. But what do plants know? Aware of through observation, inquiry, or information. They know a lot. They collect a lot of information. And in fact, the CIA claimed that a plant had witnessed the murder by stomping of another plant, could pick out the killer from a lineup of 6 suspects. So that actually comes from a book called The Secret Life of Plants, which I have to say, I actually have a copy of that at home. I didn't buy it because I really wouldn't want to spend money on it. And I wish I'd brought it in because it's a hysterical read. Anyway, Michael Pollan wrote a really nice story on it in the New Yorker. If you want a more up-to-date version of senses and things like that, this book from Chemowitz, covers more of the topic in a real-world explanation. And I haven't read the book. I've read some précis of it, but it's an interesting thing to look at if you're interested in senses and plants, which is not really what I'm going to talk about. What I want to talk about is analogies. Economics as an analogy of understanding how plants work. I think Thomas Carlyle, a Scottish economist, described economics as the dismal science, which is a very well-known commentary. But its most concise definition is how society uses scarce resources. And one of the things that we've done in the past I'll talk about is how a crop uses scarce resources. And I've just highlighted some of the words from a glossary of economics from A to F. And you can see that there's a few words in there that relate to how we think about how plants work. And I thought, well, I'll have a go at this from a psychological point of view. And of course I failed immediately because it talks about the human mind, and I guess plants don't have minds, but we conclude at the end. And I highlighted from A to F. I figured that there aren't too many addicted plants, although maybe there are. I mean, maybe some plants are addicted. I don't know of any bipolar plants. Delusional plants. Some of David Jordan's material looks a bit delusional at times. Some of the very wild crosses that he makes. But I think if from a psychology viewpoint, you're thinking about how does a crop behave to use the resources in a given context. Or that's what I've taken it to mean. So the economics analogy we've actually used before, and Eric's here somewhere, Eric Van Oostrum. Graham and Eric and I wrote a paper for a conference in Corralbin a long time ago. I think David was there, where we talked about economics of drought stress, and it made a good story, and we probably should have published it somewhere else rather than on the CD. But it was really about applying the market economy concepts of supply, demand, and equilibrium to understanding plants. And it works rather nicely. I think even people who haven't studied economics sort of have learnt this one in high school. That you can think about quantity and price being related for a given supply and demand. So if you've got this supply on the right-hand side, and that's your demand curve, and the demand curve doesn't change, you'll have that price and that quantity. And if the supply changes, it will shift the price and quantity in the appropriate directions. And you can think about that as we did in the paper, thinking about how plants respond to drought stress. So if you've got a stress starting to cut in, your leaf area is already built up. This is your light interception against leaf area index. You've got your expansion stopping. The plants are trying to shed demand by changing leaf angles and cutting their extinction coefficients. They'll start to shed leaves after the supply of water, that is, or light. Sorry, this is light. In fact, if your radiation drops down, they'll start to shed leaves and try to react and adjust to that supply. Anyway, we can give you copies of this. It's highly cited paper by us anyway. Because when I searched for it, I found heaps of hits for it. And I went, who's citing this? And it was us, anyway. So we really should publish it somewhere people can find it, Eric. If we think about psychology, the cognitive approach is the way that, the modern way, I suppose, that the mind is thought about as an information processor. And there's a lot of discussion in the literature of biological systems having been information processors, and you can get deeply buried in all of that. But the key points are some memory that allows you to store information and use information, some perception that turns that into some other signal, and attention, deciding what to do. You know, do you grow your roots? Do you grow your leaves? And it kind of sounds like a plant. The psychology of drought stress. So I tried to summarize this in one diagram, and you can go back and have a look at a chapter I wrote for a quantitative genetics book a few years ago, and Graham and myself and a few others wrote for Trends in Plant Science in 2006. And we were, I guess we weren't mentioning psychology when we did that, but what we were trying to do is think about how do plants in this field get to be the way they are because of the management that we've imposed on them. So this is sorghum, 2-row sorghum grown with skip rows or double skip. That's bare ground in there, and you think, well, that from a traditional sense and a European agricultural view, that's a big waste of energy or a big waste of resource because you're not intercepting the light that's in there, you're not using it at all. But in a cropping system in Western Queensland, those plants are growing roots underneath, and they're using the water from underneath that area. And in fact, if they do cover all of that, they'll end up water stressed because they'll run out of water. And we'll come back to that later. So how does that plant there get to be suited to this system? And that plant is reacting to its neighbours because it's been planted at a high density. And so it's been forced, it's packed in, and it's reacting to its neighbours through genetic signals which are suppressing the amount of tillering to about the right amount to adapt to this system. So there'll be some tillering, but not too much. Anyway, that's sort of, I guess, the background loop to all of this is the thinking that's kind of behind how we're trying to do our building of models, its construction of models. Now, I'd like to draw attention to some people who, there's probably a few people, in fact, I'm certain there's a few people in this room who may not really know about the famous AJPP journal, the Australian Journal of Plant Phys, which CSIRO renamed Functional Plant Biology so we could get lots more hits. And this is a really great paper from Tom Sinclair and Mervyn Ludlow. Merv was my PhD supervisor, a fantastic physiologist, very well known to a lot of people here, I believe. Unfortunately, he had a stroke at an age just a little bit after mine, and he was never able to work again, which is a very sad thing. But he and Tom wrote this great paper called Who Taught Plants Thermodynamics? And what they were trying to do was make the point that physiologists were spending a lot of time trying to measure leaf water potential in the 70s and the 60s and the 50s, and I did too. And their point was really that the plants are trying to balance demand and supply to be about zero, because otherwise the plants will explode or they'll desiccate. And the argument was you didn't have to spend too much time on leaf water potential. But the thing I wanted to pull out of it was this, which I've elaborated a little bit, but these stages of growth that, or response that Tom and Merv elaborated on back in '85, which was actually when I was finishing my original degree. And these stages that they describe about with supply being bigger than demand, you can think about supply as what the roots can supply in terms of water, and demand being what the radiation is trying to suck out of the plants. You're going through these different stages of responses where transpiration or demand is being driven by evaporation. It's being matched by root uptake or supply in this case. The RWC stays high, leaf expansion continues. As the demand goes above the supply, so you're getting more radiation load for what the roots can access and supply. The stomata are responding, and eventually they're closing. They're limited by epidermal conductance, and leaf expansion stops, and so on. So you can see that these steps are actually quite relevant to the ones I mentioned in the economics diagram. And in fact, if you go back and read that paper, the basis for a major chunk of what's in our APSIM models in terms of demand supply actually comes from that thinking from that paper. And if you want to know the description of that, it's in the sunflower paper that I wrote with Graham and Holger Meinke when I was here in my first job, actually. So, who taught plants the fables of psychology? I thought I'd better get back on my topic a little bit. The hare and the tortoise, slow and steady. The ant and the grasshopper. I don't know how many people know about that. That's about the ant saving resources for the winter, while the grasshopper's hopping around going, what are you doing? You should be enjoying summer. The Arabidopsis and the cactus, a very famous fable, solving world hunger. I guess what I want to do is talk about whether plants believe in fables and go through some of the evidence for whether they do believe in fables or not. So the case I was making before with the example from Sinclair and Ludlow is that water demand and water supply are key in the environments that we work in. We're not often radiation limited, we're usually water limited. And water demand is affected by these traits, which are combinations of both genotype and management in sorghum. And supply, which of course is the rainfall, how much that soil can hold, where the roots are, and how we manage that as well. So management's affecting both sides of that. And in fact, genotype is affecting both sides of that in different ways. And the timing of everything. So just some background for this. I mean, I know some people here might not be crop scientists. Maybe you work in livestock, or maybe you work on plants in labs and not out in the paddock. But what we're trying to do in agricultural situations is we're aiming for production. We want to use as much water as we can, as efficiently as possible, through the optimal combination of genotype and management. We're not really trying to survive. So we're not interested in being a cactus, because cactuses can live forever, but the reason they can live forever is because they don't use any water. They just get whatever water they can and they hang on to it and they don't do anything. They sit there. And that's not how you can produce a crop. The scales, and I'll cover some of these, hourly scales, daily scales, seasonal scales, generational scales. So breeders are operating in generational scales trying to accumulate genetics, because sometimes you are studying germplasm and trying to say, well, why doesn't that thing do the optimal response? And you grow maize and sorghum together, and the sorghum will beat the maize in a lot of environments. And that's because the maize hasn't got a hope. It hasn't got the genetics that'll take it there, because it just doesn't have the right combination of leaf and water management traits. These are captured in a simulation model, and I'm going to talk about different scales. I think the people who are associated with QAAFI would largely be familiar with simulation models like APSIM. APSIM's a cropping systems model with crop modules that capture all of the physiology of the system. And I've just illustrated here the water dynamics of APSIM, and these are the things that account for water supply and water demand and are the basis for the next part of this talk, how these sources of water supply and demand interact with each other. So it runs on a daily time step. You define the soil, and the weather supplies the water. And all of the dynamic emerges from the interaction of weather, soil, crop. So if we want to look at some scales, adaptation and drought stress at the leaf to canopy scale. I've gone back to another paper of Tom, Graham, and Eric, in fact, in 2009, looking at maximum transpiration rate. So this is a vapor pressure deficit during the day, that dotted line there. And this is an estimation of transpiration rate following that VPD, So this is in a situation where the roots are not limited. And what we're talking about is control at leaf and canopy level. And if you assume that there's some reaction of the stomates that will not allow this transpiration rate to go over a certain amount, say 0.6 millimeters per hour, for example, you'll drop that down and slow down that total transpiration for the day. I mean, the consequence is that your plant will heat up, and potentially that can have some negative effects, which we don't model perfectly yet, but we're working on that. And this will save water. So you'll conserve water during the hottest part of the day because you'll save water when the VPD is at its highest and it's trying to suck the most of the water out. And the consequence of this, if you run a simulation, is that the black one here, which is saving some water, so it's got that kind of response, is saving a little bit of water all the way up to flowering, and it's got some water at the end of the season. So in a terminal stress situation, it's a useful adaptation. In a situation where there wasn't terminal stress, it might not be a useful adaptation. And you'll see in some cases where the yields are high, this is just yield of the standard genotype here, and this is a relative yield. This 0.4, this limited transpiration rate, is a negative effect on yield when you've got high yields because you're not using all the water. You should have been using that water to run at maximum speed, use up all the radiation you were getting. But it does have advantages in years where you're water limited, particularly at the end of the season. Anyway, Tom's done a lot more work on that. This is a really active area of crop physiology research, plant physiology research. If you look for maximum transpiration rate, you'll probably find about 20 papers in the last 3 or 4 years, because a lot of people are studying, I think, how many papers now? 4 or 5 species at least, crop species that people are trying to look for genetic variation for this trait. And it seems like it's a useful trait in water-limited environments. And so the winner is the ant in this case. So apparently the plants believe in this fable of the ant and the grasshopper because they're saving water and they're using it later. Okay, another example I wanted to use is work that we did with Corinne Chenu when she was a postdoc at INRA. And so one of the things that we did was we took this model of leaf elongation rate, which came out of work from Francois Tardieu's lab, on limiting on an hourly basis how quickly the leaves would grow in response to temperature, VPD, and soil water potential. And so this was inserted inside of APSIM and allowing us to have that kind of response because we didn't have that level of sensitivity in the model at the time. And the rest of this is sort of APSIM kind of stuff. And we did a lot of work connecting genetic parameters to this, which you can look at in the genetics paper. So to try and keep the story simple, which I haven't because I've put one of my panels up there, so I guess I should apologize immediately. Under high VPD, go for growth, because this is a high VPD environment, so we've constructed this environment. These are the yields of all possible genotypes that you could create with these 3 traits of A, B, and C. And I think to keep it a bit more simple, the highest yields were found for genotypes that had a high A, so rapid response to temperature. They grew fast. They were insensitive to high VPD, so they just ignored the high VPD and kept growing. And they had a low response to soil water deficit as well. They just kept growing. So these plants were, I guess, the hare. They weren't the tortoise, they were the hare. And they won in this environment. Now, in some other environments, they didn't win. But in this case, the hare won, which, of course, as you know, in the fable, the hare didn't win. Now, if we then take this to group psychology, how do these things behave together at the regional scale? I've already been through this slide, so it's just to remind us about demand supply being the basis. And just to summarize a lot of different things, if you look at the combinations of maturity tillering and configuration, as in single skip or solid, so solid planting or single skip planting. You can compare all of those over 100 years at one location. So this is a slide from Graham's talk at the International Drought, in Perth last year. The one-to-one line is just the standard. And if you go for a high input genotype, so you have higher density, solid plant, late cultivar, so you're going to go in there and try and burn as much water as possible, as fast as possible, then you'll get benefits in years where the yields are high, so the standard is high, and you'll have costs in years where the yields are low, and those yields are associated with high or low water. And if you go for a really low input strategy, so you drop the density right down, you put in a skip row, you make the maturity medium instead of late, then you'll really flatten out that response. So if you're a risk-averse farmer, this is the kind of genotype you want to be growing, or this kind of input. If you're a risk taker, you want to take this one. In this case, the risk taking is not that bad. There are going to be some years that are pretty disastrous, but it still, on the whole, may well be better to take that one. So if we looked across the whole sorghum region, and sorry, I had a map of this a little bit later, but it's about, I guess, how long? 1,000 kilometres north to south. And you took one standard management, so you took plain old 50,000 plants per square metre and solid. What's the best genotype given all of this? And so you can look at this frontier of failure versus yield and select out these genotypes and say, well, what do these genotypes represent? What are the types that are in there? And in this case, the attributes of those are that they're early to medium maturity, medium to high tillering, narrow root angles. So I haven't gone through all the detail of the background of what genetic traits we looked at, but these are three of the things that were looked at in this work. And what if your management is changed? So let's think about 1 year, and I won't explain all the dimensions in detail on here, but this is yield, this is earliness or lateness, and this is tillering. So this is high tillering on this end and low tillering near the axis. And if you change the management, you'll change the profile. So this three-dimensional diagram is showing the entire genetic capability of the breeding program as we know it. So this is all of the diversity that David's got to work with for the traits of tillering and flowering time, averaged out in two different managements. And you can see that the genotypes that are optimal for a solid plant in terms of tillering and maturity are not the same ones as for a double skip. But unfortunately, that's for 1 year, and the story gets worse because, well, actually, this is the full landscape. I only showed you a little bit of the landscape. I haven't got time to explain all of this, but that was for 2 traits. This is for 4 traits at once. And the red parts are high yielding, the yellow parts are low yielding. So you can imagine that this is for 1 year, for one set of management. If David could grow all of his genetic diversity, if he had it all, because he doesn't have it all anyway, he's only got some combinations of these, he's got random dots scattered across this. That's the kind of yield landscape he could see in those 2 types of management. But then it gets worse, because if he did that 10 years in a row, it would start to change. So the breeders in these environments are working on a moving landscape. So you can see that red flipping from one side to the other across these panels. I think all you need to take away from this is that that's showing you that in each year at one location, the optimal genotype is a very different combination of traits. And this is allowing us to explore that. It allows us to do a lot more regarding which are the ones that are most often to be useful, and to try to work out how do we guide the breeding program towards those spaces. But it's really important for people who haven't been exposed so much to the variability of Australian environments and cropping systems to really have this at the back of your mind. Because, we go out there and we plant 2 or 3 trials, or even we do 3 years of trials. And we think, well, we've got a really good understanding of our system, and you really haven't. The system is a lot more complicated than that. One way of looking at this is to say, well, if I took those frontier genotypes, what are their traits? And if you look across all of the regions at once, so this is the whole region, the genotypes dominated by ones that have shallow, is that shallow Vijaya? I always forget the 30 and the 50. They're that way, aren't they? They're narrow, that's right. That's what I thought. They're the deep roots. I get them backwards sometimes. So if you look across all of these, the best genotypes on that frontier tend to have the deep roots. They've got an early to medium maturity and probably one to two tillers. If you go to different regions, you'll see that those distributions change. So this is the composition of those genotypes that are on the frontier, and it will change with the region. Now, that's a function of what management is used in those regions as well as what environments those regions experience. So we take this one step further and we look at simulating specific adaptation. And if you look at each of these, you can say, well, compared to the global standard, what happens if I look just at CQ West? I get high density, lower tillering. So these are the shifts in density or maturity and tillering. So either a management shift in density or a genetic shift in tillering or in maturity that gets me on the global optimum. And the winner is the hare or the tortoise or the ant or the grasshopper. So I guess maybe plants don't believe in fables. I still have 10 minutes left to entrap you. So I thought I better chuck in a couple of slides on the things that we're trying to do to address the next, I don't know, make the next step in this work. And I guess the things that challenge us at the moment is how do we better understand the dynamic of leaf to crop? How do we get a better idea of what's going on over the day, over the season? And the one that really perplexes us, and that's why we're grateful for people like Vijaya who like digging holes and understanding roots, is how do these allocation rules work? How do we understand how plants invest in the future infrastructure? And I guess I fell into economics again, but anyway, to capture resources and make use of those. And one of the things we want to do to capture that is to understand these weekly and seasonal dynamics of growth, and of course remote sensing approaches are part of being able to do that. I guess I couldn't resist putting this in just to amuse David for a minute. What's limited by genes and physiology? Is a crop less optimal because of the company it keeps? So is it just your neighbours that make you underperform? You know, is your office surrounded by underperforming people as a plant? Is it the company it keeps? Is the crop less than optimal because it just doesn't want to perform? It's probably not part of the story, or because it can't. And I think because it can't is actually a really useful question, because a lot of the time we think, oh, it's just genetic diversity. We haven't got enough genetic diversity to make it perform. And in fact, a lot of the time we've got the genetic diversity, we just can't get the genes in the right combinations because breeders have so many priorities to address that there are, and again, some of you people might know this, think this is obvious, but it's good to understand how breeding and genetics works, because there's so much linkage drag of chunks of genome every time a breeder makes a cross that a lot of the times they're maintaining structures and combinations of genes that produce a trait value that never gets investigated. And it doesn't get investigated because they got priorities of breeding for quality or breeding for disease or breeding for something else, and they can't break that up. And a lot of the time we immediately think genetic diversity is the important thing, but just being able to recombine properly is very challenging. And that's why there's a lot of interest in things like MAGIC and NAM populations to try to do that at the same time as introducing that genetic diversity. So I guess some of the technologies that we've been playing around with, and we're not new at this, and we're certainly not the first. There's been a lot of work done in this kind of area. But I guess what we're really interested in doing is, for a start, trying to monitor diurnal dynamics with things like this wireless canopy sensor. So the High Resolution Plant Phenomics Centre in Canberra are supplying these now as a national facility. So if people want to use these, they can contact them. These are monitoring every 10 minutes the temperature, and it's all wirelessly transmitted back. And we've had, well, 1 year's experience, I guess, with those so far. We're interested in trying to monitor temperature through imagery, either close up or from further away. And we're also interested in trying to understand this water content, and Fernanda's doing a lot of work on this. This is some unpublished work of hers, but looking at trying to predict the water content of the biomass. So trying to get at that thing that Sinclair and Ludlow were talking about is, you know, how do we predict the water content? I mean, a lot of the time as physiologists we ignore fresh weight because we always want to know dry weight, but we're not going to get dry weight out the field. We're going to get fresh weight, so we're going to have to understand how that works. The last thing I thought I'd mention as a technology is the aerial imaging. So I'm sitting on a paper at the moment that I'm just trying to finish the second set of reviews in this obscure journal. It was an invited paper, so I didn't have much choice. But we spent a fair bit of work on an internal project to get this helicopter working in a way that would allow us to do really regular measures of crop cover, diurnal canopy temperature, and to my surprise, estimates of 3D lodging. So this wasn't something I anticipated doing. We got a mosaic of images. We sent them off to some guys in the US who managed to stitch them together in a commercial service. They sent them back, and I looked at the data and went, oh, I can estimate lodging from this. And in fact, we can do a really good job of estimating the amount of lodging and the percentage of lodging through analysis of these images, because every time the helicopter takes a photo every 3 seconds, it's producing a stereo image. And the thing that we thank for that is that the plants don't move while we take the photo. If the plants were moving, of course the stereo wouldn't work. So the stereo imagery that I've got of myself or anybody else in the paddock doesn't work so well, because usually we've moved in the 3 seconds. The last point I want to make on this was in trying to turn this into in-season phenotyping. Again, Fernanda's got a lot of interest in this. This is just a series of images at different times of the season from the heli. For each of these, we can extract all of the cover information. We can feed that in as ground cover. We can convert that to leaf area index, predict the biomass and the water use. And so the way we've done this is working with Bang Yu Zheng, is feed the ground cover into APSIM, run it thousands of times for lots of combinations of traits, and then try to identify the traits that match some of the things that we're observing. And so we want to use this type of thing as a in-season phenotyping to get at things like water use, which you can't measure properly other than with a neutron tube. And it's all right putting neutron tubes out there, but I can tell you not many of our field staff are interested in putting in 2,000 neutron tubes and monitoring them over the season. So these are the types of technologies we want to get into. So I really just put this up as a reminder to myself to think about, well, are plants really able to be analysed through psychological analogies? Well, maybe I didn't do a very good job. I think this bit's all right. I think that they do store information, they perceive information, and there's lots of journals you can go and read plant signalling and regulation and think about how they actually work, or how people are thinking they work. I'm not a believer in plant neurobiology myself, but maybe there are some of you out there. I don't want to point fingers. But I guess what I concluded was that, well, plants don't believe in fables, so we can focus on physiology and maybe the rational mind theory of economics. In fact, I think economists would be better served by studying plant physiology, because at least a lot of their assumptions wouldn't be foul of the reality of people. So The rational mind theory, as most people know, is that people in a market react in a rational way, and of course they don't, which means that lots of economic theory is just rubbish because it just doesn't work because they're not able to model the rational mind. And so I'd actually recommend to economists that they should study plants, because plants have rational minds and they'll behave in a market for resources in a much more sensible way than people will, and maybe they'll get some new insight into their theory by studying it. And I just wanted to leave you with one more commentary from a Geelong, ex-Geelong supporter. This is Mark Jackson, 1985. I'm not going to sing the words. I think you all know them. If we had enough beers, you could do it. But Jacko claimed that he kept an open mind because he was thinking all the time. And I wanted to conclude by saying that plants are individuals and you can't fool them. So, thank you.